好，第8題就有關於基礎遺傳學嘅題目啦。今次咧，我哋研究嘅咧就係粟米粒粒。咁我哋有4款啦 ，A、B、C、D。咁啊，佢哋分別咧有啲係黃色，有啲係紫色，有啲咧就巢掹掹，有啲咧就好順滑嘅表面。咁呢段文字都有解釋到啦，點解佢會有紫色，點解會有黃色咧？關鍵嘅咧就係有冇一款色素啦，有冇個 pigment 啦。如果佢有嗰個色素嘅，就會顯示成紫色；如果佢冇嗰個色素咧，嗰個粟米粒內在嘅組織，佢本身嘅顏色咧就會顯現咗出嚟，就即係黃色啦。而嗰個順滑嘅粟米粒同埋一個巢皮嘅粟米粒咧，原來係建基於入面儲啲咩嘢食喎。呢一 part 咧，大家留意咯。綠色呢一句説話咧係題目俾你嘅。咁有陣時書本真係學唔嚟噶嘛。咁題目俾咗啲資料你就好好咁運用佢啦。佢就話啦，一個順滑嘅粟米粒咧，入面儲存住嘅係一啲澱粉質，而一個巢皮嘅粟米粒咧，入面就儲存一啲糖分，佢係一啲甜嘅粟米同埋澱粉嘅粟米係有分別嘅。而嗰個粟米粒粒咧，幾時為之成熟啊？就係當佢乾嘅時候就為之成熟啦。而嗰一款甜嘅粟米粒咧，當佢乾咗嘅時候，佢就會變咗巢皮啦。咁 part A 就問我哋咯，根據翻滲透作用嘅概念，就要我哋解釋下啦。點解一個甜嘅粟米粒粒，佢乾咗之後會變咗巢皮，而一個入面儲存澱粉質嘅粟米粒咧，即使乾咗都仍然係順滑呢？咁呢條題目呢，就考我哋3樣嘢嘅。第一就係澱粉同埋糖有咩嘅分別。第二，乜嘢叫做滲透作用啊？而第三，一個粟米粒當佢成長嘅時候，佢會經歷啲咩嘅過程呢？咁我哋逐步嚟解構咯。咁右下角嘅呢2幅圖呢，就係想話俾我哋聽，喺一個甜嘅粟米入面，佢啲細胞儲存住嘅係一啲糖分。而一個澱粉嘅粟米嘅細胞入面儲存住嘅就係澱粉質。咁呢個 concept 最關鍵嘅就係究竟邊一樣嘢係溶到落水啦？糖呢係溶到落水嘅，但係澱粉質呢係溶唔到落水嘅。咁有咩咁特別啊？就係由於糖係溶到落水嘅關係，佢就會將個甜嘅粟米入面嘅細胞嗰個液泡入面嘅細胞液呢個水勢就會降低咗。相反啦，雖然澱粉質係儲存咗喺個細胞入面，但係由於佢係唔溶響水嘅，所以佢唔會影響呢個粟米粒嘅細胞入面嘅水勢嘅。咁第二個概念咯，滲透作用又點解啊？滲透作用就係形容一個水嘅正向移動，由一個高水勢嘅位置去到一個低水勢嘅位置，而當中係需要一個差異滲透嘅薄膜，亦即係我哋嘅細胞膜啦。咁所以呢一度咧，你要留意咯。喺一個甜嘅粟米嘅細胞入面有一啲糖分，佢就會令到個細胞嘅液泡啦，或者佢嘅細胞質啦，佢嘅水勢就會下降，而低過出面，呢啲水就會正向入咗去個細胞，就從而就令到個細胞咧變得較為膨脹一啲啦。相反啦，一個儲住澱粉質嘅細胞咧，咁雖然入面係好多澱粉，但係佢唔影響水勢啊嘛，所以係唔會有一個正向嘅水嘅移動嘅，所以亦都唔會有滲透嘅出現啦。跟住啦，就去到最重要嘅概念啦。當一粒粟米粒當佢成長嘅時候啦，究竟經歷啲咩階段呢？當佢成長嘅時候，佢會吸水。咁啊，因為水對於一個細胞嚟講非常重要。以前我哋學過噶啦，對於個細胞質入面有唔同類型嘅化學反應係發生緊嘅，而水就係一個媒介。同一時間，水亦都係一個溶劑，去俾唔同嘅溶質去溶咗落去，如糖分啦、礦物質啦，都要溶落水嘅。當個粟米粒要成熟嘅時候呢，佢就會乾咗。而乾咗嘅時候，本身一個脹卜卜嘅細胞入咗好多水。而家當佢一失水嘅時候呢，由於粟米粒嘅外層呢係冇彈性嘅，所以當佢啲水流失嘅時候，佢唔會變翻做平時順滑嘅樣，而當初脹咗嘅部分呢，拉扯咗嘅部分呢，就會收縮而變得巢皮啦。好，去到 part B， 我哋就研究下個粟米粒咯，佢嘅顏色啦，佢嘅形狀啦，呢2款特徵呢，原來都係由唔同嘅基因所去控制，而呢啲基因呢，都係喺唔同嘅染色體上面嘅。嗱，呢個資訊非常之關鍵。咁啊，因為我哋喺書本學緊嘅呢，都係呢個情況，就係基因之間呢，係冇一個連鎖嘅關係嘅。所以啦，控制粟米粒粒顏色嘅基因，同控制粟米粒粒佢個形狀嘅基因呢，係一個獨立嘅狀態嚟嘅。咁下面呢幅圖咧就講啦，我哋有2款純種嘅粟米，純種嘅紫色同埋順滑嘅粟米粒，另一款就係純種嘅黃色同埋巢皮嘅粟米粒。咁當佢哋經過呢個交配之後呢，出嚟嘅第一代 F1 呢， 就全部都係紫色同埋順滑嘅粟米粒。題目就問啦，基於成個交配嘅結果，叫我哋推論一下邊一款嘅表現型係顯性呢？究竟紫色係顯性定係黃色係顯性呢？究竟順滑嘅粟米粒定係巢皮嘅粟米粒係顯性呢？咁成條題目啦，就係考翻我哋4步成詩啦。如果大家唔記得嘅話，快手睇翻呢2段片啦。一開始我哋就要理解下一個家長嘅表現型，從而推論佢哋嘅等位基因究竟係乜嘢。第二啦，就睇下我哋嘅後代啦 ，offspring 佢哋嘅表現型，跟住又去估下佢哋嘅等位基因嘅配搭咯。跟住啦，到第3步就非常之重要啦。喺成個家庭入面，有冇任何一隻嘅生物，佢哋係集合型嘅生物呢？當有集合型嘅生物嘅時候，我哋就可以好順利去到第4步啦。究竟邊一個嘅等位基因係一個顯性嘅等位基因，或者係一個隱性嘅等位基因啦？咁而家逐步嚟擊破咯。第一步就係阿爸阿媽都係純種，即係咩意思呢？即係話佢哋只係能夠產生到一款嘅配子啦。咁你會見得到啦。如果黃色又好，紫色又好，順滑又好，巢皮都好，佢哋都係屬於大 A、大 B、細 A、細 B， 甚或乎中間呢2個情況，大 A、細 B、細 A、大 B， 佢哋都係純種。喺呢度咧，梁 Sir 想提一提，好多同學咧可能誤會咗嘅，淨係以為咧前面呢個大 A、大 B 同細 A、細 B 先係純種，其實中間呢2個都係。因為咩叫純合，咩叫集合啊？個理解就係講緊喺同一款特徵底下，佢哋嘅等位基因係一樣嘅。即係話啦，當我哋想去睇呢個粟米粒嘅顏色嘅時候呢，咁其實佢 either 紫色或者黃色啦。咁只要佢嘅等位基因係一樣，無論係大 A 定抑或係細 A， 咁其實佢已經係一個純種嘅狀態。只有大 A 加細 A 呢個先叫集合型嘅狀態。所以就要提翻大家。前面咧 A 咧我就當係粟米粒嘅顏色啦，而後面個 B 咧就係粟米粒嘅形狀啦。咁其實你睇粟米粒嘅形狀，細 B 同大 B 又何嘗唔係純合呢？係咪？咁所以啦，當我哋知道咗呢個概念之後呢，我哋就會知得到佢哋嘅後代必然係一個集合型。嗱，呢個情況呢，我哋擺咗喺單基因遺傳呢都係一樣嘅啫，即係話啦，大 A 乘細 A。佢哋出到嚟個後代一定係大 A 加細 A， 一定係集合型。而有集合型之後，我哋最重要嘅就係能夠指得出佢個基因型同埋表現型嘅關係，從而就會知道邊個係顯性嘅表現型啦。所以咧，答案就逐步嚟咯。第一，當我哋知道咗阿爸阿媽都係純種，即係話啦，所有嘅第一代都係集合型。呢個就係頭先我講嘅第3步有咩咁重要啦。因為當我知道咗邊一個係集合型之後，我哋就會有紫色嘅呢一句最常見嘅說話啦。喺一個集合型嘅情況底下，只有顯性嘅等位基因能夠被表現出嚟，而隱性嘅等位基因係會被遮蓋咗，表現唔到出嚟嘅。所以我哋會發現啦，既然全部 F1 都係集合型，而全部 F1 都係出到紫色同埋順滑嘅粟米粒，所以紫色同埋順滑嘅粟米粒就係一個顯性嘅表現型啦。佢 B2 呢，又去到另一個 cross 啦，我哋用翻 F1 頭先我哋講嘅集合型啦，咁佢就同翻一個純種嘅黃色同埋巢皮嘅粟米粒做一個交配。頭先我哋喺 B1 已經知道咗，喺呢個紫色同埋順滑嘅粟米粒呢，佢哋係顯性。咁即係話呢個黃色同巢皮嘅粟米粒呢，就必然係隱性咯，係咪啊？佢要解釋下啦，點解 F1 同埋呢一個純種嘅黃色巢皮嘅粟米粒去交配之後呢，出到嚟啦，嗰個結果會係咁樣嘅呢？又有紫色，又有黃色，又有順滑，又有巢皮嘅呢？好亂喎！我哋就要睇一睇呢條題目考緊我哋嘅係乜嘢啦。成條題目就係考緊我哋嘅 Mendel's Second Law 曼德爾第2定律獨立分配定律。一個基因嘅等位基因嘅分配係獨立於另一個基因嘅等位基因嘅分配，亦即係正頭先題目講過俾我哋聽噶啦。控制粟米粒顏色嘅基因同控制粟米粒形狀嘅基因，佢哋係處於不同的染色體之上。亦即係話佢哋嘅分配必然係獨立噶啦。所以喺我哋嘅答案入面呢， 都一定要提翻 Mendel's Second Law。咁除咗概念之外呢，題目都考我哋一啲技巧嘅。第一就係數據分析。今次個結果係點樣呢？我哋喺第二代，我哋見到一啲紫色嘅、黃色嘅、巢皮嘅、順滑嘅粟米粒，我都見到喎。第二啦，就係嘗試利用基因遺傳圖或者一個龐氏表，去將一個集合型即係 F1 同埋一個純合型嘅隱性嘅生物去做一個交配。而佢第三就係利用文字去描述呢個遺傳圖同埋龐氏表。呢一 part 你會有個問題啦，梁 Sir， 你係咪唔識英文啊？呢度咪寫咗咯，你用基因遺傳圖去作答係唔會俾分噶。啱啊，我知啊，我識英文噶，畫基因遺傳圖當答案係冇分啫嘛，有冇話扣你分啊？冇啊嘛。咁但係點解梁 Sir 仍然要畫 genetic diagram 俾你睇啊？其實你都可以噶。個比喻就好似當你個心算未係咁叻嘅時候，你咪寫條直式俾自己去計數咯。2乘4就係2乘翻個4。咁啊，你他日日子有功啦，你咪知道2乘4等於8咯。但係之前冇嘅，畫出嚟，就算你畫落個答題簿嗰處啊，唔通佢交叉你跟住減你4分咩？唔會噶嘛，頂多唔俾分你嘅啫。但係你透過畫咗呢個基因遺傳圖，你再用文字去描述，咪就係題目想考你嘅嘢咯。所以其實考都係考嗰啲嘢㗎咋，你係識噶，不過你只係將圖表化成文字啫嘛。嗱，畫呢幅圖其實就好快嘅啫，你就唔使好似梁 Sir 畫得咁複雜嘅，因為佢今次唔係答案嚟噶嘛，今次只係草稿嚟嘅啫嘛，係咪？就用 G 咧就形容個粟米粒嘅顏色，而家 T 咧就係形容個粟米粒嘅巢唔巢皮同埋順唔順滑啦。咁我哋發現啦 ，F1 咧就係大 G 細 G 大 T 細 T。而純種嘅黃色同巢皮呢，就係細細 G 細細 T， 係咪？跟住啦，畫翻啦，有幾多款嘅配子啊？佢哋嘅第二代，佢哋嘅基因型同佢哋第二代嘅表現型又如何啊？逐一擊破啦，咁多位。而家我哋就拎住呢個遺傳圖，用文字好好咁表現佢出嚟啦。第一係一個純種隱性黃色巢皮嘅粟米嗰樹，佢只能夠產生到一款嘅配子，啱唔啱啊？你見到噶嘛？然後啦，第一代嘅集合型生物，佢哋就會產生到4類型嘅配子，係因為個等位基因係獨立地分配 ，assorted independently， 亦即係曼德爾第二定律獨立分配定律。佢嘅答案其實都唔係咁好嘅，因為最好係應該講埋 according to Mendel's Second Law， 用中文嘅話即係依據曼德爾第二定律。我哋就睇得見啦，原來呢，第一代嘅集合型係會產生4款嘅配子，而呢4款嘅配子就會經過一個隨機嘅受精作用，就會產生咗4款嘅基因型出嚟。而呢4款嘅基因型正正就係我哋嘅 A、B、C、D， 紫色得嚟係順滑，紫色得嚟係巢皮，黃色得嚟係順滑，黃色得嚟係巢皮。呢一個嘅做法係適合一啲你對成個 genetic diagram 個空間感未係最熟習嘅同學去理解嘅。但係當你慢慢熟嘅時候，你會發現啦，F1 擺到明就係集合型啦，集合型咁咪4款配子咯。咁隔離係純隱性啦，咁即係得1款配子咯。咁溝埋有幾多可能性啊？畫一畫出嚟就搞掂㗎啦。你嫌基因遺傳圖慢嘅，咪畫龐氏表咯，都好快嘅啫，係咪？所以呢一個 part B2 呢，我想話俾大家聽就係識變通啊！你畫真係冇蝕底㗎，係只有幫咗你嘅啫。咁你話哇，係咪花好多時間？呢幅圖你認真畫嘅話，半分鐘一定畫得完。跟住去到 part C 啦，其實就係有關於科學本質嘅題目啦。咁所以大家之前如果未睇呢段片嘅話呢，就快睇啦。每年呢都有呢款題目㗎，無論係 MC 定係長題目。但係唔係個個人攞得足啲分嘅。當初阿 Mendel 咧，佢去發表啊，當啲生物咧佢遺傳嘅時候，啲特徵係點樣去遺傳到落去㗎？喺嗰一個時代咧，其實啲人係唔知咩叫染色體嘅。咁究竟啦，阿 Mendel 係點樣得出呢個假説嘅咧？咁其實當中啦就係第一，佢搜集好多嘅數據啦。而基於佢所得出嘅數據嗰啲咩3:1啊、9:3:3:1啊、300幾隻、500幾隻嘅後代啊，之後咧佢就透過邏輯嘅推敲去嘗試解釋佢所見到嘅結果啦。呢度咧，我想同大家分享一個有關於科學本質嘅説話。呢句説話梁 Sir都好中意嘅，就係科學或者科學家佢哋唔係知道晒所有嘢， 係啊，啱㗎，我哋真係唔知啊。但係正正就係因為我哋唔知，所以我哋先會繼續向前進發，而唔係固步自封。知道晒咧，搞掂晒咧，解釋晒所有嘢咧，咁跟住就停滯不前。亦都借助呢句説話去鼓勵大家終身學習，學多啲嘢。好，跟住啦，又嚟到第8條嘅一點出發啦。嗱，成條題目咧考我哋3樣嘢嘅啫。第一就係滲透，第二就係雙基因遺傳，而第三啦就係科學嘅本質。成條題目咧就由個粟米粒咧去出發嘅，當中要考我哋嘅就係滲透啦，同埋雙基因遺傳啦，佢嘅顏色啦，佢嗰個質地啦，有啲咩嘅唔同咧？而題目再追問我哋嘅就係獨立分配啦，有關於孟德爾第二定律啦。今次嘅題目就冇考我哋畫遺傳圖，但係下次可能考我哋咧。但係啦，大家點樣學都好啦，基因遺傳圖就要識得畫，如何去描述個基因遺傳圖都要識得去講。OK， 咁去到第二個啦，就係有關於滲透，就考翻你有關於個溶質嘅濃度啦，同埋個水勢。今次嘅題目咧，你會覺得非常之 genetics 啊。但係下次啦，佢純粹就係問一棵植物嘅繁殖，咁啊得唔得？嗰個粟米粒咧有啲咩咁重要啊？我放落泥土嘅時候咧，佢會唔會發芽噶？佢啲嘢食係從邊度而嚟噶？其實都可以問你嘅。而有關於成條題目係遺傳學嘅發展咧，亦都符合咗科學嘅本質啦。所有嘢係 keep 住改變嘅，好多嘅發現咧都係基於我哋一啲新嘅發明，一個科技嘅進步，例如顯微鏡咯。例如啦，一啲更精密嘅、更高倍數嘅顯微鏡就幫助到我哋去觀察染色體嘅行為啦。咁我希望每一次一點出發咧，你都會知道呢條題目究竟考緊啲乜嘢，同埋亦都知道究竟他日溫書可以點樣溫。In question 8, we are going to study the appearance of some kernels of a corn. So according to the diagram, you can see 4 types of the appearance of the kernels: A, B, C, D, purple or yellow, smooth or wrinkled. And in the paragraph, it also explains that why the kernel of the corn will appear as purple or appear as yellow. You can see the green and the yellow highlighted sentences, so they are related to the part A. And for the whole question, I would like to tell you that somehow we do not learn everything from the textbook. And there are some basic information, crucial information provided in the question. For example, the pigment of the kernels and the shape of the kernels. The smooth kernels store starch, while the wrinkled kernels store soluble sugars. And the surface of the sweet corns becomes wrinkled when the corns dries up. So for part A, with reference to osmosis, explain why the kernels of the sweet corn becomes wrinkled when they dry up, but the kernels of the starchy corn remains smooth. So in this question, there are 3 main concepts to recall the difference between starch and sugar. The second one, we need to recall the definition of osmosis. And for the third one, the development of the kernels of a corn. So in the diagram below, I show you the cells of the sweet corn and the cells of the starch corn. So you can see that the cells of the sweet corn, they store the sugar in the cell sap of the vacuole. And the cells of the starchy corn, so they store the starch in the cytoplasm. So first of all, you need to recall the difference between starch and sugar. Each one is water soluble. So the sugar molecules, they are water soluble. And for the starch, they are water insoluble. And the key idea is that which one will lower the water potential of the kernels. So the sugar molecule will dissolve in water, and the sugar concentration will be very high, and the water potential of the cell sap in the vacuole will be lower. So that's why there will be a net movement of water molecules from the region of high water potential to the region of lower water potential across the differentially permeable membrane. So we can see that during development, the kernels of the sweet corn will draw more water by osmosis than those of the starchy kernels. And the third concept, we need to recall the development of the kernels of a corn. First of all, they will absorb the water. So for water, we learned the importance of the water before. They act as the solvent for different chemicals to dissolve in, and they provide the medium in the cytoplasm for different chemical reactions. So at the beginning stage, the kernel will absorb water. But after that, at the mature stage, they will dry up. Therefore, when the kernels dry up, the kernels of the sweet corn, they will shrink as the result of losing a lot of water. And as the outer layer of the kernel is not elastic, so the kernel of the sweet corn becomes wrinkled. So you can see that for the whole question, new ideas which you may not learn from the book is the last 2 points only. But for the first 3 points, you learned it before. Starch and sugar. Can you tell me the difference? Which one is water soluble? Which one is not water soluble? And then can you link this idea to the osmosis? Or once something they can dissolve in the water, so it will lower the water potential of that particular solutions. So that's something you can learn. And then you can tell me any net movement of water molecules takes place. After that, you may need to deduce the answer logically. And for part B, we are going to talk about the basic genetics. So there are 2 traits of the kernels are controlled by the genes located on different homologous chromosomes. So that's why those genes, they are not in linkage. And the following diagram shows the result of a cross between 2 purebred corn plants. Purebred purple smooth kernel and the plant with the purebred yellow and wrinkled kernels. And in the first generation, the plant, they have the purple and smooth kernels. So based on the result of the cross, that means you need to take a look at the first generation and deduce which phenotypes are dominant. So in this question, we are checking the 4 steps of the pedigree analysis. If you have not yet watched these 2 videos, watch it first. For the step 1, we need to identify the parent's phenotype, purple, smooth, yellow, wrinkled, and deduce the possible allele, that means the possible genotype. And for step 2, we need to take a look at the offspring's phenotype and then deduce the possible allele or the genotype. And for step 3, which is very important, identify who is or are probably the heterozygous individuals. And for the last step, we can determine the dominant and recessive allele. And for the scaffolding, you need to state the parent's phenotype and genotype first. Both of them are the purebred, so that's why they will only produce one type of gamete. So you can see from this table, for the big A, big B, small a, small b, you can see from this table, all of them are the purebred organism. So they can only provide one type of gamete. And one thing I would like to remind you all is the definition of homozygous and heterozygous to clarify your concept. Some of the students, they may think that big A, small b, or small a, big B, they are not homozygous. They are heterozygous. They are not purebred. They are purebred. Because for the homozygous, the definition is that same allele for the same trait. For example, we are talking about the color of the kernel, a purple or a yellow. So for the big A, homozygous. And for small a, also homozygous, same allele for the same trait. But what is heterozygous? It means that different allele for the same trait. For example, I'm still talking about the color of the kernel. So what if the genotype is big A, small a? So it is heterozygous. You can see that all the F1 will be heterozygous. And then for the 4th one, you need to recall the critical concept between heterozygous genotype and the phenotype. And finally, we can determine the dominant phenotypes. So let's see how can we construct our answer. Both parents are purebred. It's given by the question, right? We do not need to make up the whole story. And then the F1 produced are all heterozygous. That's something we learn about the genotypes and phenotypes. And for the purple one, in heterozygous condition, dominant allele will be expressed while recessive allele will be masked. So that's the critical concept we need to recall. Therefore, purple color and smooth surface of the kernel are the dominant phenotypes. So you can see from the result, all the F1 corn plant, they contain the purple and smooth kernel. So that's why purple and smooth kernel, they are the dominant phenotype. And for part 2, the F1 organism, so they are crossed with the purebred yellow and wrinkled kernels. And you can see the result. There are purple, yellow, smooth, and wrinkled kernels. So for F1, very quick reminder. So let's recall the part B1 result. Purple and smooth kernel, they are the dominant phenotype. So that's why for the yellow and wrinkled kernels, they are the recessive phenotype. So we need to explain the result, of course, using Mendel's law of inheritance. So now we are talking about two types of traits. One is color and the other is the shape. So obviously, the critical concept is the Mendel's second law, law of independent assortment. It states that the separation of allele for one gene is independent of the separation of alleles for other genes during gamete formation. At the very beginning, it was mentioned that the genes controlling the color and the shape of the kernels are located on different chromosomes. So that's why you have to recall the Mendel's second law. And the critical skills of this question we are checking, first of all, data analysis. You need to use the result of the cross. So you have to mention the second generation. There are purple, yellow, smooth, and wrinkled kernels. And the second one, you need to draw a genetic diagram or a Punnett square, which is talking about the heterozygous organism crossed with the homozygous recessive organism. And then use wordings to describe the genetic diagram and Punnett square. At this moment, you may ask, Mr. Leung, do you know English? Can you read this? Marks will not be awarded for genetic diagram. Yeah, I see it and I know it. And I do it intentionally to explain it to you. The metaphor is talking about if you cannot do the mind calculation, so you need to write down the formula to help yourself, right? And it is the same idea for drawing the genetic diagram to help you to do the description part. The idea is that even I draw the genetic diagram, even I try to want it to be the answer, I may just get the 0 mark. It was not mentioned that marks will be deducted. It's just talking about marks will not be awarded, right? So I have this genetic diagram to help you. The big G, small g refer to the allele of the purple and the yellow kernel respectively, and the big T, small t for the shape. So you can see that I have the F1 and the purebred. I draw the genetic diagram in 30 seconds. You do not need to follow the complete version of the genetic diagram because it is not the answer, right? It's just a draft to help you. After drawing this, the next step, we can observe the genetic diagram or the Punnett square to describe it by wordings. First of all, only one type of gamete will be produced from the purebred compound with yellow and wrinkled kernels. Yes, I see it. And the individuals in F1, they are heterozygous with genes for two traits located on different chromosomes. It was mentioned by the question. I do not need to make up the story. Therefore, the allele for the trait are assorted independently based on the Mendel's second law. The suggested answer by the HKEAA is not perfect. I have to mention it is the concept from the Mendel's second law. And therefore, 4 types of gametes will be produced from the F1, and after random fertilization, the zygotes with 4 different combinations of genotype will be produced. Yes, I can see it from my genetic diagram. And then it leads to the expression of all possible phenotypes A, B, C, D in the F2 generation, which means the purple smooth, purple wrinkle, yellow smooth, and yellow wrinkle. I see everything from the genetic diagram. So for this part, I just need to tell you that you need to be flexible to answer the question. Don't just see the words that marks will not be awarded for the genetic diagrams and then I do not draw it. Draw it. Why not? Even you draw it as the answer, just 0 mark. But after you draw it, it helps you to write your paragraph. And for part C is the question about the nature of science. So what if you have not yet watched the video of nature of science? So watch it now. And every year there must be questions asking the nature of science, no matter long question or MC. So for part C, when Mendel proposed how traits are inherited, chromosomes had not yet been discovered. In your opinion, how did Mendel come up with his hypothesis? So the idea is that, first of all, he collected a lot of data of 300, 500, 800 of the plants, or what is the color of the beans, or what is the shape of the beans, or whatever. And then based on his observation on the number of offspring with different phenotypes. So he worked out a possible explanation for the observation by logical deduction. So we can see that science knowledge is based on and or derived from the observation of the natural world. And science is a process of ongoing inquiries. We keep asking questions and keep doing experiments to observe. And finally, we know that scientific knowledge is also tentative and subject to change or modify. The past, we did not know anything about chromosome, but now we know something more. So we can keep modifying the law of the genetics. And I would like to share with you a quote about the nature of science. People keep saying science doesn't know everything. Yes, we don't know everything. And science knows it doesn't know everything. Otherwise, it would stop. So I would like to use this quote to encourage you all for lifelong learning. Keep learning, keep exploring to new things to enrich your life. For question 8, I would like to talk about the overall concept checking first. This question is about 3 aspects. First aspect, osmosis. And the second one, dihybrid inheritance. And for the third one is the nature of science. For this question, it just starts from a kernel. So for the kernels of a corn, it's checking 2 concepts. First of all, dihybrid inheritance. And for the second one, osmosis. So for the dihybrid inheritance, is checking the concept of Mendel's second law. So what about next time I can ask you the Mendel's first law? And for this question, it doesn't ask you to draw the genetic diagram, ask you to describe the concept. But maybe next time it will ask you to draw the genetic diagram. So you need to refer to the video before. So how can we draw the genetic diagram? So for the osmosis, is checking the idea of solute concentration and the water potential. For question 8, it focuses on the genetics a bit more. Maybe next time it may ask you from the aspects of plant reproduction. The whole question is about the development of genetics and is related to the nature of science. Especially, we have the advancement of the technology. For example, we have the microscope to observe the chromosome behavior. So I hope that this curriculum mapping can help you to do the revision.